In the previous video, we discussed the importance of the molecule of DNA and how is a long molecule that is going to be found in the nucleus with important function. We also mentioned that the molecule of DNA is going to be wrapped about histones, and more than that, that the function of the molecule of DNA is to make proteins. Now we're going to look at what are the components that are going to help us understand how DNA can replicate, that means to make a copy of itself, and also how it can send the information to the ribosome by the molecule of RNA. The first component that we will find in any nucleic acid will be a sugar unit. That will be a pentose. Let me put that in writing. A pentose, that could be two different ones. A ribose that we have seen before is an aldose, and in this case we need to look for having the beta anomer. That means the H is above the overall plane of the ring, and also that is the ribose is the sugar unit that is going to be found in the molecule of RNA, and when we have two hydrogens on position number 2 of ribose, that will be the one called 2-deoxyribose, and that is what gives the name to DNA molecule, deoxyribonucleic acid. Now, when it comes to the sugar units found in nucleic acids, we are going to change instead of the numbering, this is the anomeric carbon number 1, say anomeric. We are going to call those with the prime number, and the reason is that the nitrogen bases that are also part of the molecule of DNA will carry the plain numbers, like the pyrimidines and purines will carry the numbers 1 through 9 for the purine, and from 1 to 6 to the pyrimidine, but the sugar units will carry the prime numbers. Now we say that the molecule of DNA will contain deoxyribose, the RNA molecule will have the ribose. Now the nitrogen bases, we also have a difference between the two different kinds, and that is the pyrimidines. One of the pyrimidines that will be part of DNA, in rare cases you will find uracil as part of the molecule of DNA. That will meaning that it is actually, if you find an uracil in the molecule of DNA, it's likely to be a thymine that has been damaged. If you see the difference between those two, the only difference between thymine and uracil, thymine being part of DNA and uracil being part of RNA, is just a methyl group. So one uracil in DNA is probably a thymine that has been damaged. Now, all others will be part of both DNA and RNA. We have adenine being part of both, guanine, cytosine. All these three belong to both DNA and RNA. Now, the combination of a sugar unit plus a nitrogen base is what we're going to call a nucleoside. For example, when I combine adenine with the anomeric carbon of ribose, I'm making what is called adenosine. Adenosine, indicating that now this is a nucleoside. And we have one on top and using the same one. So we can see that ribose, using its anomeric carbon, is going to react. So we're going to remove a molecule of water, and that's going to make a beta nitrogen glycosidic bond. Now, the molecule of DNA will contain what is called the phosphodiester bonds. How is that made? First, we need to introduce, we have difference between what is a nucleoside and what is a nucleotide. A nucleoside, we say that it is the nitrogen base plus the sugar unit. In the nucleotide, a nucleoside, so this is a nucleoside because we already have a nitrogen base and an OH. So a nucleoside that can react with a monophosphate, diphosphate, or triphosphate that will become a nucleotide. So nucleoside, sugar, let's say here, a nucleoside, that will be the sugar plus the nitrogen base. Sugar plus the nitrogen base, that is a nucleoside. A nucleotide is a sugar plus the nitrogen base plus a phosphate unit. We have the three components for a nucleotide, and that's actually what I'm showing in here. So we have the sugar unit, the nitrogen base, that is an aromatic nitrogenous base, and a phosphate unit that will be a nucleotide. But once we have nucleotides, we need to make a polymer of nucleotide. Remember, the introduction of this chapter was reminding you that we have seen polymers of glucoses to make, let's say, starch, or polymers of amino acids to make a protein. Now we need to make a polymer of nucleotides. So we will have one nucleotide with a free hydroxyl group, like in this sugar unit, that is going to react with the phosphate unit of a second nucleotide, and that is to make a connection between the two sugar units. So we will have a connection between two sugar units, but that will not be just a simple oxygen like it was for when we have the glycosidic bond, but that will be a phosphodiester bond. That will be a phosphodiester bond to connect the two sugar units. And of course, these sugar units are connected to nitrogen bases. That is important to remember, that this reaction that we have in here is similar to what it was when we have the combination of a carboxylic acid end and an amino end. We have two different ends in a nucleotide. You have a hydroxyl group end, like in this one, and you also have a phosphate end. So when you combine two of them, that has to be with what is called an activated nucleotide. If it's an activated nucleotide, that means that the deoxyribose is connected to 3 phosphate units. So we are going to let go, or let's say release, what is called pyrophosphate. That is 2 phosphate units. So we are releasing 2 phosphate units, and that is an SN2 reaction that we're going to look in more details later on. Now, continuing with the components on how the molecule of DNA will work, we will say that this molecule will have what is called a primary structure. So that will be similar to what we have seen before. We say in amino acids and proteins, we say that the primary structure of a protein was the sequence of amino acids. Now we have to create what is the primary structure of this nucleotide, or what is the backbone. Let's say the backbone is what repeats in this polymer. The backbone, what repeats in this polymer is what? We are going to repeat a phosphate unit that is combined to a sugar unit, that is another phosphate unit, to another sugar, phosphate, sugar, phosphate. That is going to repeat. That is going to be constant. And remember that we have 3 meters of a primary structure. And then the last one will have a free hydroxyl group. We see a free hydroxyl group and connected by a beta glycosidic bond. This sugar unit will be connected to a nitrogen base. In this case, that will be a guanine. The next one is a thymine, and this one is the last. Let me put the last here. This is an adenine. I'm gonna put some more in here. Let's say this is a G, and then let's say that this one is a C. So we have a sequence. What is the backbone is what repeats. What is changing is what is the nitrogen base that is bonded to the sugar unit in the N-beta glycosidic bond. So we have that the sequence of the nitrogen bases is of extreme importance. In the chapter of amino acids and proteins, we said that the R groups that were attached to the alpha carbons were bringing versatility to a protein. They were bringing some hydrophilic amino acid side chains or some hydrophobic amino acid side chains, so that determined how the protein was going to fall to make alpha helices or beta sheets. Now the sequence of these nitrogen bases is going to determine what will be the sequence of the molecule of RNA that is going to determine what will be the sequence of the amino acids to put together during protein synthesis. Let me repeat myself. So we have a sequence of DNA. Let's say this one is a ATG, and we're going to put the phosphate end, that is the 5' end that has a phosphate unit, is free, and that's of extreme importance. And the G has the 3' end, that's a free OH, all right? And then we're going to say that this is the molecule of DNA that is going to determine what will be the sequence of RNA. And that sequence of RNA is going to determine what will be the sequence of amino acid side chain that will tell me what will be the tertiary structure of the protein or the quaternary structure of the protein. Let's say here the 3D structure. The sequence of the amino acids is going to help me to determine if I will have alpha helices, beta sheets, or if I'm going to have a globular protein, an inside out, or if it's going to be a fibrous protein. So that is of extreme importance, but remember what's the sequence. We're going to say that the genetic information is going to flow from DNA to RNA and from RNA to protein. And that is, we're going to have what is called from DNA to RNA. That process is going to be called transcription. You are sending the information of nucleic acid phosphodiester bond continues as nucleic acids with phosphodiester bond, and from the molecule of RNA that brings phosphodiester bond to peptide bonds. So we now know that we have a backbone, but the molecule of DNA is not a single strand. If we look at the differences between DNA and RNA, we're going to start by saying, first, we understand that we are making phosphodiester bonds, and that is going to happen for both, for DNA and RNA. And that is nothing but the combination of a hydroxyl group and a phosphate group. That will be an SM2 mechanism, so you will have to have an activated nucleotide, so that means that is triphosphate. So let's say that this is an adenine. If I have an adenine, it has to be an ATP, deoxy ATP molecule, because I will need to have a good leaving group. So I will have a nucleophilic attack as an SN2, and then I will have these 2 pyrophosphate leaving, and then I will be creating a phosphodiester bond. This is a phosphoester on that direction, and this is also a phosphoester in that direction. If you remember, we have an ester, which is this bond, and now we have a mixed ester, because now we have a phosphorus ester for 2 carbon 3' and also 2 carbon 5' of the second sugar unit. Now let's see. We said that in the molecule of DNA, this is going to be a double strand. In the molecule of RNA, it's going to be single strand. So we say, yes, it was here a double strand, but we're just not presenting that. That's coming in the next. But the molecule of DNA has to be a double strand. We will see that this molecule has is anti-parallel. So if in one end I had the 5' end of the phosphate unit, if in one end I had the 5' end of the phosphate unit, on the other end I have the 3' OH. So when I have the second strand, this one will be here 3' OH. Let me put the OH here. And on that one, that will be a phosphate unit. So you say that is anti-parallel because the sugar units are running on this side is like upside down. And then you will have 5' parallel to the 3' end. So one difference between DNA and RNA is that in DNA you have a double strand. We usually call that DS. And in RNA you will have single strand. Another difference that we will have is the sugars. Deoxyribose for DNA, ribose for RNA. What's the difference between those two sugars? Ribose, as we know from the chapter of carbohydrates, has OHs in all carbons, as when it is an aldose and it's a pentose. Deoxyribose is made from ribose by an enzyme that is called a reductase that removes the OH, and now we have, instead of having 2' with OH, 2' lacks oxygen, so 2' has a carbon with 2 hydrogens instead of the OH. Now the other difference is that the pyrimidine thymine is not present in RNA. This is the one that the only difference between those two is a methyl group. So we say yes, RNA also has phosphodiester bonds, but we have replaced thymine with uracil. And the other guanine and cytosine will be part of the molecule of RNA.